After death, the physical body ceases to function, but the fate of one's consciousness or soul remains the subject of diverse beliefs and speculation. But what if there is a wake-up after the death on future? The Project Cryonics. Cryonics is an effort to save lives by using temperatures so cold that a person beyond help by today's medicine can be preserved for decades or centuries until a future medical technology can restore that person to full health. Cryonics sounds like science fiction, but is based on modern science. In more defined terms, cryonics is the practice of preserving a deceased person's body or brain at extremely low temperatures, typically using liquid nitrogen. The purpose is to essentially halt the biological decay process and keep the body or brain intact for an indefinite period of time. The procedure involves several steps. First, immediately after legal death, the body or brain is rapidly cooled using cryogenic techniques. This is done to minimize damage to the tissues and organs that can occur due to lack of oxygen and blood flow. Next, cryoprotectants, which are substances that help prevent ice crystal formation and cellular damage, are introduced into the body or brain. These cryoprotectants act as a type of antifreeze, protecting the delicate structures from damage during the freezing process. Once the body or brain has been cooled and treated with cryoprotectants, it is carefully placed in a specialized container or vessel designed for long-term storage. These containers are typically stored in cryonics facilities, which are equipped with advanced technology to maintain the extremely low temperatures required for preservation. The hope is that in the future, advancements in medical technology and understanding of the human body will allow for the revival and restoration of these cryopreserved individuals. It is believed that if the tissues and structures can be preserved without significant damage, there may be a possibility to reverse the conditions that led to death and restore the person to life. However, it's important to note that cryonics is still considered an experimental procedure, and its success and feasibility are subjects of ongoing scientific research and debate. The field faces many challenges, including the potential for cellular damage during the freezing and thawing processes, as well as the complexities of reversing the underlying causes of death. When it comes to cryonics, cryoprotectants play a crucial role in preserving the body or brain during the freezing process. These specialized chemicals are introduced to the tissues to prevent ice crystal formation and minimize damage at low temperatures. The cryoprotectant solution is typically perfused or circulated through the blood vessels, replacing the natural fluids within the tissues. This helps ensure that the cryoprotectant reaches all parts of the body or brain, including the cells. The choice of cryoprotectant depends on various factors, such as its ability to prevent ice formation, its toxicity level, and its compatibility with the tissues. Commonly used cryoprotectants include glycerol, dimethyl sulfoxide, DMSO, and ethylene glycol. These substances have unique properties that make them effective in reducing cellular damage during the freezing process. Before introducing the cryoprotectant, the body or brain is cooled gradually to a temperature where ice formation is minimized. This is typically done using a process called vitrification, which involves reaching a state where the tissues become glass-like rather than crystalline when frozen. Once the optimal temperature is reached, the cryoprotectant solution is slowly introduced. This allows for the gradual replacement of the natural fluids with the cryoprotectant, preventing damage to the cells caused by sudden changes in osmotic pressure. After the cryoprotectant has been fully introduced, the body or brain is ready to undergo the final cooling process and be transferred to the cryostat for long-term storage. It's worth noting that the choice of cryoprotectant and the specific protocols used can vary among different cryonics organizations or facilities. Research and development in this field are ongoing, with the aim of finding even more effective cryoprotectants and improving the overall cryopreservation process.